Since we already mentioned the changes that can happen based on pH and temperature, it's not surprising that that's one means in which we can regulate enzymatic behavior, the rates of enzymatic reactions. But can you also envision how you can regulate the reactions by the concentration of substrate versus product? So if you've got plenty of substrate, but you have a ton of product, it's like you have nowhere to put what you're finishing, so that will slow the reaction down. Likewise, if you have a ton of substrate, you're going to be making a lot of product. It just depends on how fast you can push the reaction in one way or another. But there are three other ways that we can regulate enzymes. From a competitive nature, allosteric regulation, which we will get into, and it's not as bad as it sounds, and then covalent modification. So let's take these sort of one at a time. So our competitive inhibition, and our example are going to be drugs, such as, for instance, penicillin. But what happens is when you have enzymes that normally bind to a certain substrate, that substrate specificity, and when you do, it processes them and you end up with the products that are released, inhibitors actually are able to also bind to the active site. But they bind in such a way that they can't be processed. You can't form that transient transitional state. And so they sort of get stuck, and it prevents the natural occurring substrate from binding. And so you slow down, or depending on the concentration of your inhibitor, you completely stop the natural occurring reaction. And that's a competitive inhibitor of an enzyme. You see how it works? It just blocks it and overwhelms the system by having so much of this other material. Now, allosteric. I want you to understand that allosteric means we're not doing anything to the enzyme at the active site. That competitive inhibitor, where was that inhibitor binding? The active site. Allosteric means a different site, a different place on the enzyme. And I want you to think of the allosteric site as the on-off button for a machine. I'm working on refurbishing my deck back home, and so I use a table saw. There's the business end of the saw, that's the blade, but then you've got that on-off button in some other place. So the allosteric site is an on-off switch for the enzyme. And the active site's the saw blade. Can you think of it like that? So in our example here, we are going to have, this is our enzyme. The substrate comes in and binds to the active site. It gets processed through the induced fit. Products are formed and released. But you see this other slot above it? If we have an inhibitor, an allosteric inhibitor, that means this molecule comes in and binds and turns the enzyme off. And it turns it off by changing the shape of the enzyme and changing the shape of the active site such that the substrate can no longer bind. So some enzymatic conditions can have an enzyme that's on until an inhibitor comes in and binds to the allosteric site to turn it off. Now, can you already envision the opposite? Let's have an enzyme that's off normally and naturally until we have an allosteric activator come in and bind, changes the shape, but now it changed the shape to the active site, which now is available to bind substrate and process it into products. You see how that can work? So the allosteric site is an on-off switch. It depends on the enzyme and it depends on the allosteric partner if it's going to be an inhibitor and turn it off or an activator and turn it on. Did you have a question?
Did you say it changes the shape of the enzyme and the substrate?
Well, no, the active site. See, I mean, the whole enzyme, but But really, where we're focused is the active site. You see, we've got this one that doesn't fit until the activator comes in, makes it perfect, just like the natural, normal on switch, and we can process. So that's allosteric regulation, either inhibit or activate. But allosteric means a different place on the enzyme. Now, one really kind of cool, smart way of allosteric regulation is feedback regulation. Now, when you get into physiology, we're going to see that your body and your normal physiology works basically on negative feedback. Negative feedback means whatever information you're putting in, the output is the opposite. It's like your thermostat at home. Too hot, that's the information coming in. What is your air conditioner going to do? Cool it off. Too much heat, you take the heat out and make it cooler. So in this sense, with our feedback inhibition, here we have an enzyme that's taking, in this case, threonine, and we're producing this product, isoleucine, through various steps with different enzymes. It's a metabolic pathway. But this enzyme that started the process of taking threonine and converting it into isoleucine has an allosteric site for an inhibitor, and guess what the inhibitor is? Isoleucine. So the more isoleucine you produce, the less threonine you're going to start to break down. Does that make sense? I mean, that's smart, right? If you have plenty of isoleucine, is there a need to continually break down threonine? No. But what happens when you start to run low on isoleucine? Now you can no longer inhibit the enzyme. It's going to start back to processing threonine into more isoleucine. Is everybody? And see, that's an allosteric type of inhibition, but the product is the actual inhibitor of the initial enzyme.
So once it starts getting lower numbers, it'll kick that
It'll turn on.
and kick it out.
And once you're producing plenty of this, it'll shut it off. That's pretty cool. That's an example of negative feedback that you're going to see all through physiology of the human body. And in this case, we're talking about allosteric negative inhibition. Now, the last way that you can modify enzymes is covalently. Can you already imagine how this is going to be a permanent change? This is not going to be an easy on, easy off change. You're going to have enzymes to add things and enzymes to remove things. And in some instances, once you process it, you can't go backwards. And so we're going to see phosphorylation and dephosphorylation, adding a phosphate group and taking a phosphate group off is a means with which we can modify materials, predominantly proteins. And in fact, in physiology, when we look at the contraction of smooth muscle in your blood vessels, smooth muscle in your GI tract, the ability of smooth muscle to contract is dependent upon myosin, one of the contractile proteins along with actin, but it's dependent upon myosin being phosphorylated. And the enzyme that does that is myosin light chain kinase. A kinase is an enzyme that adds a phosphate. You have to add a phosphate to myosin to get it to unfold so the contraction can happen. But to get that myosin to fold back up so the muscle can relax, you have to pull that phosphate off. There's a different enzyme, myosin light chain phosphatase. So this is how you can modify materials, you can modify proteins, and it changes the physiology of what's going on. Another way is proteolytic cleavage. What do you think proteolytic literally means? Just decipher that word. Proteo, protein, lytic, lysis, breaking. So you cut pieces of the protein off. And so here is a phosphorylation, dephosphorylation. So what we're doing is we are taking some phosphates off of ATP and we're converting glycogen, glucose and glycogen, we're moving that back and forth. We're going to see through the processing of glucose. We're going to add phosphates, and we're going to take phosphates off throughout that whole thing. That's enzymes adding and taking away. Proteolytic cleavage, here's our pancreas again that we mentioned a little while ago with trypsin. When this material is first processed and injected into the duodenum, the first part of the small intestine, these enzymes are called pro-carboxypeptidase, chymotrypsinogen. Anytime you hear pre or pro or ogen at the end of a word, that means it's in an inactive form. And you have to cut off portions of that protein to convert it into the active form. And I like to say you have to cut the ogen. If you've got chymotrypsinogen, cut off a piece of that protein, remove the ogen. Now you've got chymotrypsin. That's the active form. And it's not just digestive enzymes. You have to do the same thing with the filament that is responsible for causing you to form a blood clot. Those are pretty important, wouldn't you say, when you cut yourself? You have an inactive filamentous protein circulating in your plasma right now called fibrinogen, and you have to cut off, proteolytically cleave off that protein to convert fibrinogen into fibrin, which is the sticky spiderweb-like filament that's going to cause everything to stick to it and form a clot. But I think you can imagine, once you cut off the protein, are you ever going to get it back on? No. That enzyme or that protein is done. You got to make more. So covalent modification in some instance is a permanent change, especially this proteolytic cleavage one. We said enzymes are largely proteins, and they are, but just to make you aware, we do have some enzymes we could say are RNA molecules. And we can loosely apply this to ribosomes, because ribosomes are going to be made up of ribosomal RNA. And there are a few other examples, so we can't say all enzymes are proteins. Most. You see how it's a very powerful word? Most. You get close to saying all, but most, except for, because there's always going to be some exceptions. So some of our enzymes are in fact made up of RNA.